Hi, my name is Jillian Murray, and I'm going to be walking you through my cellular metabolism poster. So starting up in the corner, we're using the production of a glucose molecule to produce a molecule of phosphoribosyl pyrophosphate, which will be used later in the process. We're also able to use glucose in the glycolysis pathway to yield 2-pyruvate. This pyruvate is going to go through the pyruvate dehydrogenase complex, where we produce 2-acetyl coenzyme A and 2-NADH. These molecules are essential for the citric acid cycle, where using our acetyl coenzyme A, we're able to go through this process with all of these intermediates and make 6-NADH, 2-ATP, and 2-FADH molecules. These intermediates are produced by the production of amino acids. We're also able to take the FADH and NADH molecules that we produced via the citric acid cycle to go through ATP synthase, where the subunits of the ATP synthase complex pump protons from those high energy molecules that we created to make more ATP. It does this by making proton gradients back and forth between the membrane using other coenzymes and cytochrome C. This allows us to make even more ATP from the original ATP that we invested way back in glycolysis. So with all of this ATP produced and different subunit molecules, we're able to go through this process of making nucleotides and more amino acids. One more just subunit that makes an important thing, we're able to break down glycerol via glycerol kinase and produce glycerol 3-phosphate, which we'll need later in the process. But starting up here, we need to make orotate in order to make uridylate. So we're able to take a carbon dioxide molecule, which we produced earlier in the pyruvate dehydrogenase complex, and an amine molecule, and go through this process to create orotate. Orotate is then the start of the process to make uridylate. We're then going to use uridylate to make our essential nucleotide triphosphates, UTP, CTP, and TTP. We have to do the same thing in order to make ATP and GTP, except we have to make inosine. So with that molecule of PRPP that we produced way earlier in the pathway, we're going to be able to input that and go through this whole process to make inosine, which will then be able to be catalyzed and broken down into ATP and GTP. And this is how we make our 5 essential nucleotide triphosphates, which are the building blocks of our DNA and our nucleotides, and the foundation of all of our genetic material. We're also able to create some really important steroid molecules for the membrane. So using the glycerol 3-phosphate that we produced over here, we're able to go through this process to make triglycerols. And then we're able to initiate the mevalonate pathway, where we're able to produce dimethylphosphate, and using dimethylphosphate, we can produce a molecule of squalene. Squalene is essential for membrane function. It keeps the membrane together and holding shape, and cells would not be able to live without squalene being part of their membranes. This is really important. And we're also able to use extra glutamate molecules and acetyl coenzyme molecules that we produce during the citric acid cycle or pyruvate dehydrogenase. We're able to use this and add a little bit of the ATP that we produced from ATP synthase, and we're able to go through a process to make arginine, an essential amino acid. We're also able to do the same with 3-phosphoglycerate that we produced earlier in the cycle. We're able to go through another synthesis pathway to make serine and glycine. Phenylalanine is also able to be transferred into tyrosine via phenylalanine hydrolase. And then we're able to make methionine by going through more of a process and inputting some of the other amino acids that we produced earlier, like serine and adenosine, in order to make molecules like cysteine, and then get another molecule of succinyl coenzyme A out of this. So via just one glucose molecule, we're able to produce a ton of ATP, and then use that ATP and our intermediates to produce important intermediate molecules that then can catalyze the production of our essential nucleotide triphosphates and all of our amino acids that we need in order to function as a cell.